I would like to introduce our first speaker. It's a big pleasure for me to introduce Alicia because she's a colleague and a very good friend. Alicia is a Brazilian biochemist. She's full professor at the Institute of Chemistry in the University of São Paulo. She graduated as a in medicine at the University of Campinas, where she made also her PhD, and then she made postdoctoral period in the Oregon Grad Institute in the USA. Her research focuses mostly on mitochondrial bioenergetics, redox biology, and how metabolism, diet, and mitochondrial ion transport interact with chronic diseases and aging. She has an impressive scientific production in these areas, and I would like to mention that she was awarded in 2024 with the L'Oréal UNESCO Prize for Women in Science. And we are very proud also running together an initiative. In fact, she's leading the initiative of building up a new center at the university, which is called Center of Metabolism. The acronym will be COMETA. So this is something that is making us very proud. So without any more words on my side, I would like to thank Alicia, and to give the microphone to her. Thanks a lot.
So thanks, Ariel, for this really interesting invitation to speak in this initiative here. Let me see if I know how to share my presentation. Oh, apparently I'm at the end of my presentation already here. Now let's go to the beginning.
Yeah, you are in edition mode now.
Is that correct?
Yes, now yes.
You can see my screen, everything's working. So
yes.
Thank you again for inviting me. I think this is a really interesting initiative to try to bring quantitative measurements into metabolism. I certainly need that. And what I'm going to try to convince the audience today is that mitochondrial calcium transport is a central node in metabolism. That mitochondrial calcium transport is both sensitive to nutrient availability, so for example, dietary interventions, and that also that mitochondrial calcium transport changes can change pathways in cells that are nutrient sensitive pathways. And the way that we came upon this finding was actually studying obesity. So obesity is a big problem worldwide. It's a problem that's increasing. And though recently we have better drugs to treat obesity, they still aren't perfect for every case of obesity. And we always have to evolve on top of treatments. So we really do have to understand the molecular mechanisms behind obesity if we're going to understand how to treat this disease and also how to treat other diseases that are associated with obesity, and mainly associated with obesity and aging. And the way we do this mostly is with our favorite model of obesity, which is not what most people think of as a model of obesity. So our favorite model of obesity is actually your normal laboratory rat or mouse, your normal laboratory rodent. This is something that people don't think about often, but we rear our laboratory rodents under ad libitum feeding conditions. So our rodents in the lab are allowed to eat whatever they want, whenever they want, in whatever quantities they can eat. And although we give them a diet that is quite healthy, if you can eat as much as you want, because we are evolved to overeat, because we evolved to survive conditions of lack of food, these animals will overeat. And by the time they're young adults, by the time they're 2-3 months old, they already exhibit many of the hallmarks of an obese human. So if you look at the quantities of body fat, if you look at the insulin levels, if you look at the inflammatory markers of a laboratory rodent reared under ad libitum conditions, they're the typical markers of a mildly obese human. On the other hand, if you dietary restrict these animals or calorie restrict these animals, and we typically calorie restrict the animals in 40% of the ad libitum diet and give them a diet that has vitamins and minerals, so they have exactly the same quantity of vitamins and minerals, but they'll have 40% less calories. If you do this kind of caloric restriction, you prevent all these markers of obesity that you see in these normal laboratory mice or rats. So basically, I'm going to compare ad libitum fed animals as a model for obesity with calorically restricted fed animals as a model for normal lean human being. And we know that this is an important way to prevent obesity and to prevent diseases associated with obesity for 90 years already. So McKay and co-authors showed in 1935 that if you do restrict calories and prevent obesity, you can increase the lifespans and, very important, increase the health spans of laboratory animals by about 50%. You prevent age-related diseases and increase the life expectancy of these animals. So, how did we end up finding out that dietary interventions could change mitochondrial calcium uptake? We found this out very serendipitously, actually, when Ignacio Amigo came to the lab to do a postdoc. And he was very interested in the effects of calorie restriction and excitotoxicity. So, excitotoxicity is a form of neuronal cell death in which neurons literally die because they get overexcited. So it's exciting neuronal cells to the level of toxicity. So normally in a synapse, when you have the release of neurotransmitter, this is going to increase calcium in the receiving neuron. In excitotoxicity, the neurons, for some reason, and usually it's because of lack of energy, are going to release too much neurotransmitter. And too much neurotransmitter is going to lead to too much calcium in the receiving neuron. And too much calcium can lead to neuronal cell death. And excitotoxicity is really important in a number of neurological diseases, which are both age-related and related to obesity. So we have excitotoxicity involved in damage related to stroke. We have excitotoxicity involved in damage related to Alzheimer's disease. And we also have excitotoxicity in neuronal damage involving Parkinson's disease. And all of these diseases are less incident when you prevent obesity. So what Ignacio was asking is, does calorie restriction actually prevent all these neurological diseases through separate mechanisms? Or does the prevention of obesity prevent excitotoxicity and therefore prevent all these neurological diseases? So to answer that question, he promoted excitotoxicity very directly by injecting animals with cyanuric acid. And cyanuric acid is going to allow the activation of these receptors in neurons and the increase of calcium inside of neurons very directly. So he got laboratory mice, submitted them to 3 months of either an ad libitum diet, so they were obese, or a calorie restriction diet, so they were lean, and then injected them with cyanuric acid and looked at lesions in the brain by looking at seizures that these animals developed. And what he saw was an enormous effect of calorie restriction. So what you're looking at here is the time that these animals present in a seizure state, which is very much prevented in the calorie restricted animals. And what you're looking at here is the number of limbs that are affected by these seizure states, which tells you how serious the seizure states were. And you can see that the calorically restricted animals, when they do have seizures, they have very limited seizures, while the ad libitum obese animals, when they have seizures, they can have seizures that affect more of their body. So the first question is already answered here. It is that calorie restriction can very, very strongly prevent excitotoxicity in animals. Now, how does that happen? What is the mechanism behind that? And that's where mitochondrial calcium comes in. Because mitochondria can take up calcium, and in taking up calcium, they can prevent too much calcium in the cytosol of neurons, and therefore they can prevent excessive calcium associated with excitotoxicity. So mitochondria can take up calcium through the mitochondrial calcium uniporter, which is an inner mitochondrial membrane protein that allows the entry of calcium into the matrix. And this calcium is attracted by the negative charge of the matrix. So this is an active uptake down the electrochemical gradient in mitochondria. The mitochondrial calcium uniporter is part of a mitochondrial calcium uniporter complex. There are all sorts of accessory proteins that are associated with this protein. And it can take up calcium with lower affinity than the ER, but much, much higher capacity than the ER. Mitochondria can actually take up enough calcium in the matrix that it precipitates in the matrix. So very large amounts of calcium can be buffered by mitochondria. Now, once calcium enters mitochondria, mitochondria can extrude calcium either in exchange for protons, so protons enter and calcium leaves, or in exchange for sodium ions. And this is known as the sodium-calcium exchange, typically associated with the NCLX protein in mitochondria that I'm going to talk about later. Mitochondria can also take up excessive calcium. And when mitochondria take up excessive calcium, this is associated with oxidative imbalance, oxidation of membrane protein thiols, and the formation of a very large pore known as the mitochondrial permeability transition pore. And under these conditions, mitochondria lose this excess calcium, but they also lose intermitochondrial membrane integrity, so they lose their capacity to do oxidative phosphorylation. And we wanted to know, after Ignacio saw that there was this enormous prevention of excitotoxicity, if there were changes in mitochondrial calcium uptake and calcium buffering in mitochondria. So to answer that question, he isolated mitochondria from the forebrains of either calorie-restricted animals who were lean in blue, or ad libitum-fed animals who were obese in red. And then he incubated these mitochondria in the presence of calcium green, which is a calcium probe. It's a fluorescent calcium probe that does not transverse membranes. So it's going to measure calcium outside mitochondria. And when mitochondria take up calcium, it's actually not going to measure this calcium anymore because calcium is buffered inside mitochondria. Under these conditions, he does a whole bunch of additions of boluses of calcium. Every time you do a calcium addition, you see this increase in fluorescence because calcium green is fluorescing, which is followed by a decrease in fluorescence because the mitochondria are taking up this bolus of calcium that was added. And after some time, you can see that mitochondria kind of slow down in their calcium uptake. There's a decrease in this ability to take up calcium until calcium uptake stops in mitochondria. And from these traces, we can measure two things. One of them is the calcium retention capacity. It's the total amount of calcium that mitochondria can take up under these conditions. And the second thing we can measure is the calcium uptake rate, or the speed in which mitochondria can buffer calcium after the first bolus of calcium addition. And to our surprise, we saw that the exact same amount of mitochondrial protein, how did I do that? The exact same amount of mitochondrial protein could take up more calcium and could take up calcium faster when we looked at the mitochondria isolated from the calorie-restricted animals versus the mitochondria from the obese animals. So the mitochondria from the obese animals can actually take up less calcium, and they can take up calcium at a slower rate. And this explains completely this enormous protection that we see against excitotoxicity, because the mitochondria from the lean animals can buffer more calcium and can buffer this calcium faster, avoiding this increase in calcium that you see in the cytosol that leads to excitotoxicity. Now, we actually uncovered the mechanisms in which this occurs, the whole signaling pathway that leads to this increase in mitochondrial calcium uptake with caloric restriction. I'm not going to go through this because the paper has been published for a number of years. But what I think is really important from this data is that this was the first, at least to my knowledge, evidence in the literature that a dietary intervention could change mitochondrial calcium transport properties. And this seemed to me to be really important because nutrient availability is a central regulator in metabolism. Mitochondria are central regulators in metabolism, and calcium is a central regulator in metabolism. We all studied all these enzymes that are regulated by calcium when we were undergrads. So these three nodes of regulation and metabolism being related to each other seem to be really important to me. And for a number of years now, we have looked at how dietary interventions change mitochondrial calcium uptake because of that. And just for an example, the effect that we saw with calorie restriction in the brain is not limited to the brain. So this is work from Sergio Menezes' PhD. And he could see that there's also an increase in mitochondrial calcium uptake in liver mitochondria in calorie-restricted animals. And what we actually saw was that many different tissues and many different diets change mitochondrial calcium uptake. And it's actually diet-specific and tissue-specific in how these mitochondria respond. So now, what we know now is that changes in caloric intake and changes also in the way that we eat, the time that we eat, can change calcium uptake properties in mitochondria. What's the consequence of this? So we know since the 1980s that certain enzymes involved in oxidative phosphorylation are activated by calcium. And we were curious to know if this change in mitochondrial calcium could affect oxidative phosphorylation. But we wanted to look at this using modern techniques and quantitative techniques that involve measurements of metabolic fluxes in mitochondria. And this was the postdoc of Eloisa Villas-Boas in the lab, in which she did very carefully calibrated mitochondrial calcium uptake experiments, so we know exactly how much calcium is inside mitochondria. And she also looked at mitochondrial oxygen consumption rates as a measure of flux so that we could determine quantitatively how much oxidative phosphorylation was happening. She also did other non-flux measurements, which I'm not going to mention here. But basically, what she saw is what we call the Goldilocks effect of calcium on oxidative phosphorylation. So if you remember Goldilocks' story, she looks at various different things in the house of the bears, and many of these things are too much, many of these things are too little, but then she finds the just right temperature of the porridge or the just right thickness of the bed. So Goldilocks sees that there are things that can be too much, that can be too little, and that can be just right. And that's actually what we see with calcium in mitochondria. So if you look at state 3 oxygen consumption, which is oxygen consumption in the presence of ADP or in the presence of oxidative phosphorylation, if you have too little calcium, you don't have a very high oxygen consumption rate. If you have too much calcium, you don't have a very high oxygen consumption rate. But if you have just right amounts of calcium, you have an activation of oxidative phosphorylation. So you have just right amounts of calcium that will lead to the activation of oxidative phosphorylation. And this makes sense because too little calcium won't activate enzymes that are calcium sensitive in mitochondria, while too much calcium will lead to the mitochondrial permeability transition and loss of impermeability of the inner mitochondrial membrane. So just right calcium concentrations will activate oxidative phosphorylation. Now that's an isolated mitochondria. These are isolated mitochondria from liver. What happens in intact liver cells? Now, in intact liver cells, we can measure mitochondrial calcium, for example, expressing proteins that are calcium sensitive, such as this mitogecko system here. And we can change the amount of calcium in mitochondria, for example, releasing calcium from the ER with hapsigargan, or chelating calcium in the cytosol with BAPTA-AM, or either pharmacologically or genetically manipulating the MCU, which allows calcium to enter mitochondria, or the NCLX, which allows mitochondria to release calcium into the cytosol. And really interestingly, all these interventions, when we did them, would either decrease the maximal respiration, the maximal ability to have electron transport in mitochondria, or would not change electron transport in mitochondria. And what this tells us is that liver cells under normal basal conditions are exactly with their Goldilocks concentration of calcium. Surprisingly to me, they don't use calcium to upregulate oxidative phosphorylation. They're already at their maximal activation of oxidative phosphorylation at their Goldilocks calcium level. So I showed you that caloric intake can change calcium in mitochondria, that calcium in mitochondria can regulate oxidative phosphorylation. But at the same time as we were seeing this, many groups in the literature were also seeing that mitochondrial morphology and dynamics was responsive to caloric intake, so would change with nutrient availability, and also was regulatory of oxidative phosphorylation. So this is a second mitochondrial phenomenon which seems to be linked to regulation of oxidative phosphorylation and nutrient availability. And we decided to ask the question if there was a crosstalk between changes in mitochondrial morphology and changes in mitochondrial calcium. And to do this, we collaborated with Orion Shirahai in UCLA, and we studied cells with different shapes of mitochondria. So we studied cells with control mitochondria, we're going to call them here, which are basically those bean-shaped mitochondria we often see in our books. We studied cells with very elongated mitochondria because we had disrupted the fission system of mitochondria. So Orion calls these spaghetti mitochondria. And we also studied cells in which we had disrupted the fusion system in mitochondria. So this generates very small ball-shaped mitochondria, which Orion calls meatball mitochondria. And very importantly, under all these conditions, the area of the mitochondria per cell was exactly the same, but the size and shape of mitochondria was different. So we could look at how these sizes and shapes of mitochondria affected calcium uptake. And to do this, what we did was get the cells and permeabilize the plasma membrane using specific detergents, and then study mitochondrial calcium uptake within these permeabilized cells using the exact same technique with calcium green and adding boluses of calcium to these permeabilized cells. And what we saw was that size and shape actually did change mitochondrial calcium uptake. So if you look at the control mitochondria, they can take up normal quantities of calcium at normal calcium uptake rates. If you look at the cells that have mitochondria that are very elongated, they can take up more calcium and they can take up this calcium faster. And if you look at the mitochondria that are very small, they can take up very little calcium and they take up this calcium at a much slower rate. So mitochondrial morphology has a direct impact on mitochondrial calcium uptake, both rate and capacity. And I already showed you that changes in mitochondrial calcium uptake can regulate oxidative phosphorylation. But of course, if mitochondria take up calcium differently, this can mean that the cell might have different amounts of calcium. So we decided to go back to intact cells and look if these changes in mitochondrial size and shape could impact on calcium in the cell and also calcium in the ER that I'm not going to show you. So what we did here was incubate the cells with Fura2, which is a cytosolic calcium probe, and then deplete these cells of calcium and add calcium outside the cells back to look at store-operated calcium entry, to look at repopulation of calcium into these cells. And what we very consistently saw was that the cells that had very elongated mitochondria could take up calcium much faster, repopulate their calcium much faster than the control cells, the cells that had normal-sized mitochondria, and much, much faster than the cells that had very, very fragmented mitochondria. And in fact, the cells that had very fragmented mitochondria ended up having lower levels of final calcium, both in the cytosol and the ER, related to these low amounts of calcium that these mitochondria can take up. So changes in mitochondrial calcium uptake can impact on calcium in the cytosol and calcium in the ER. So we have changes in mitochondrial calcium happening because of changes in caloric intake. We have an impact of this oxidative phosphorylation. And we also have a crosstalk between changes in mitochondrial size and shape, changes in mitochondrial morphology, and changes in calcium uptake, which not only impacts on calcium in mitochondria, but impacts on calcium in the cytosol and in the ER. Now, when you have changes in calcium in the cytosol and in the ER, this changes signaling pathways all over because there is so much signaling in the cell that happens because of changes in calcium uptake. And this is basically what has occupied us for the last few years. We've been really interested in changes in mitochondrial and cytosolic calcium levels that are related to changes in nutrient-sensitive pathways. So what I'm going to show you are two stories of how changes in mitochondrial calcium can change these nutrient-sensitive pathways. And this is what I'm going to end our whole story with. And the first story I want to tell you is how changes in mitochondrial calcium can affect autophagy. Autophagy is important because it's a nutrient-sensitive pathway. So basically, autophagy is activated under conditions of low nutrient availability. And it's really important both to remove molecules that have been damaged in some way, but also to make nutrients available to cells when there's a lack of nutrient availability in cells. And in a nutshell, what happens is that there's an activation of LC3 in the LC3-2 form. This is going to congregate on membranes of autophagosomes, and these autophagosomes are then going to fuse with lysosomes, and this is what's going to promote the degradation of the cargo inside this autophagosome lysosome fusion vesicle, releasing nutrients to the cell. And we were interested in autophagy and its relationship with mitochondrial calcium transport because autophagy is regulated by many calcium sensitive points. And it's also regulated by the levels of ATP in the cell. So it's also sensitive to oxidative phosphorylation. But nobody had looked at if mitochondrial calcium transport could regulate autophagy in cells until Vitor Ramos started his PhD in the lab and basically asked if mitochondrial calcium transport could have an impact on autophagy. And basically the answer is yes. He saw this in many different manners. You can read the paper, which was published last year, but I'm going to show you just some of the data in this paper now. And very elegantly, he studied autophagy using an expression system in which the expression of a green fluorescent protein and a red fluorescent protein is linked to the expression of LC3. So if you have an induction of autophagy, you're going to have a yellow fluorescence. Now, the green fluorescent protein loses its fluorescence when you have the change of pH when the lysosome fuses with the autophagosome. So then when you have this completion of this fusion of the lysosome, you're going to have red fluorescence. And this allows us to look at both the initiation of autophagy in yellow and the completion of this fusion of the lysosome in red. And what we saw was that there isn't really much of an effect of changing mitochondrial uptake through the MCU on autophagy. That didn't change autophagy much, both when we did this genetically and when we did this pharmacologically. On the other hand, if we either silenced or pharmacologically inhibited the NCLX, which is the calcium extrusion pathway for mitochondria, we saw a very strong effect on autophagy. In other words, if we have less NCLX activity, we have less autophagy in our cells, both the initiation of autophagy and the completion, the fusion of these autophagosomes with the lysosomes. Now, interestingly, autophagy is not only regulated by mitochondrial calcium transporter NCLX by the extrusion pathway of calcium, but NCLX is regulated by conditions that increase autophagy. So calorie restriction by decreasing nutrient availability increases autophagy. And what we found was that NCLX expression is increased under these conditions of calorie restriction. So there's a crosstalk between nutrient availability, mitochondrial calcium transporters, and the regulation of autophagy. Now, cytosolic calcium does not only regulate calcium in the cell and autophagy, as we saw, cytosolic calcium can also regulate metabolic activities in many different cells. And the last story I want to tell you is how mitochondrial calcium can change cytosolic calcium and change cognitive function by changing nutrient availability in the brain. And this was studies done by João Vitor Cabral Costa, also a really, really talented PhD student in the lab. And João Vitor spent a lot of his time during his PhD under pandemic conditions locked in the lab. So he did a lot of in silico work. And he was interested in astrocytes and calcium transport in astrocytes. And he found that NCLX, this calcium extrusion pathway in mitochondria, was very, very strongly enriched in astrocytes versus neurons, versus other cells in the brain. And this enrichment of NCLX and astrocytes happened relatively to other components of the mitochondrial inner membrane, so respiratory chain components, for example, and also relatively to components of the mitochondrial calcium transport machinery. So it really seemed to be an enrichment of NCLX and astrocytes. And this happened in many different parts of the brain. And this happened if you looked at protein databases or mRNA databases, there really seemed to be a lot of NCLX in astrocytes. So what does NCLX do in astrocytes? Once we were able to do experiments again, we either inhibited NCLX with CGP, which is a pharmacological selective inhibitor, or we knocked out NCLX and astrocytes and looked at what it did metabolically. And to our surprise, we didn't see any change in oxidative phosphorylation. So these mitochondria were still in their Goldilocks level of calcium in terms of oxidative phosphorylation. But very specifically, we saw an increase in glycolytic rates and an increase in lactate production and secretion when we inhibited NCLX in astrocytes. Now, NCLX and lactate production in astrocytes is not just a byproduct of fermentation in astrocytes. Actually, astrocytes generate lactate on purpose, and lactate is used by neurons as one of their most important sources of ATP. So neurons will use lactate generated from glucose in astrocytes as a really important source of energy in neurons. And different conditions that change the production of lactate in astrocytes have been previously shown to change cognition. So we asked if changes in NCLX and astrocytes could change cognitive activity. And here we collaborated with Juan Pedro Bolaños because we are not a cognitive lab. And what João Vitor did there was generate animals that didn't have NCLX in neurons or didn't have NCLX in their astrocytes and looked at different cognitive parameters of these animals. And very consistently, he saw that novel object recognition, or the ability to notice that an object is new and be interested in this new object, was increased in animals that did not have NCLX in their astrocytes, while it was decreased in animals that did not have NCLX in these neurons. And this is compatible with other data in the literature showing that situations that increase lactate secretion by astrocytes increases novel object recognition also. So mitochondrial calcium transport changes, increasing lactate secretion can have an impact on cognition. So I hope I convinced you today that changes in calorie intake can change calcium uptake in mitochondria. The changes in calcium uptake in mitochondria can affect oxidative phosphorylation in a Goldilocks type manner in which there's a just right concentration of calcium that activates oxidative phosphorylation. I hope I convinced you also that changes in mitochondrial morphology can change calcium uptake in mitochondria, and that this can change calcium levels both in the cytosol and the ER. Because of that, when you change calcium in mitochondria and you change calcium in the cell, and this has an impact on autophagy. NCLX is a very important regulator of autophagy, and also can have an impact on substrate availability in the brain. So when you have changes in NCLX activity in mitochondria and astrocytes, you increase the amount of lactate secreted by astrocytes, and this has a positive effect on cognitive function. So I have to end by thanking both the former students, many of whom I showed here today, and our current students working on many different exciting projects in the lab. Unbelievably, the lab just turned 25, so this is not the lab. This is all our former students. We got together for a big party a few years ago, and we're excited about the next 25 years, hopefully working within our Center for Metabolism together with Ariel. And thank you for inviting me here today.